Hello and welcome to this GCSE Science Revision video for additional biology on inheritance. In this video we'll be looking at mitosis, meiosis, stem cells and how genes are inherited. Mitosis is the process where a single cell divides into two genetically identical cells.
Mitosis is the process of creating two cells from one. The first stage is that each individual chromosome copies itself.
The classic X shape.
These pairs of chromosomes then condense to form an X shape and centre around a central point called a centromere. Spindle fibres begin to spread out from two particular points in the cell and move to either pole. Once the spindle fibres have spread out from the pole, the nuclear membrane disintegrates, allowing the pairs of condensed chromosomes to be attached to the spindle fibres. The spindle fibres line the pairs of chromosomes along the centre or the equator of the cell.
The spindle fibres contract.
Next stage is that the spindle fibres contract, pulling the pairs of chromosomes apart, so a full copy of chromosomes move towards either end of the cell.
And cell cleavage happens.
Once a full set of chromosomes has moved towards either end of the cell, a new nuclear membrane forms around them and the cell splits into two individual cells that are genetically identical to the original one. These two new cells are called daughter cells.
In meiosis, sex cells are made, or gametes. A gamete or a sex cell only needs half the amount of genetic material as a normal cell.
Each cell in the human body has 46 individual chromosomes. 23 from your mother, as shown by the red chromosomes, 23 from your father, as shown by the blue chromosomes. The first stage in meiosis is that all of the chromosomes double, so you get two full sets of chromosomes. The second stage of meiosis involves pairs of chromosomes crossing over.
You get something called crossing.
At the point where they cross over, some genetic material is swapped between maternal and paternal chromosomes, which allows for greater genetic variation.
From the chromosomes from the mother pass onto the chromosome from the father.
After the stage of crossing over, pairs of chromosomes are pulled to either side of the cell. This is done using spindle fibres which spread out from the poles.
At either end.
Spindle fibres contract, pulling them further to the poles.
As with mitosis.
A new nuclear membrane forms around the 23 pairs of chromosomes
into two
and
genetically different
you get cell cleavage.
Spindle fibres start
the second stage in mitosis starts off with spindle fibres spreading out from either end of the cell.
As the nuclear membrane disappears,
the spindle fibres attach themselves to the centromere at the centre of the pairs of chromosomes.
The centre of the cells.
The spindle fibres contract, pulling individual chromosomes to either end of the cell. This causes 23 chromosomes to end up at each end of the cell. At this stage, the cells split into four, so each cell splits into two genetically individual gametes.
Egg cell which is viable.
A new nuclear membrane forms around the 23 chromosomes. We can say
During the development of an embryo, most of the cells become specialised, which means that they can no longer change into a different type of cells. But embryos contain special types of cells called stem cells. These can be grown into any other type of cell found in the body. They are not specialised. Stem cells can be removed from human embryos that are a few days old. So, for example, they can be removed from embryos that are made during fertility treatment that are not used. Stem cells can be used in therapeutic cloning. So, if you were to receive a medical treatment with the cells grown from stem cells, your body's immune system would recognise the cells as foreign and they would be rejected. But this does not happen with cells that have been grown from your own stem cells. This can be done by cloning one of your cells to produce an embryo, then taking stem cells from this. This is done by taking the nucleus out of a human cell, putting a nucleus from a patient cell into an egg cell. The egg cell is then simulated to grow to an embryo, and embryonic stem cells can then be taken from this. When questions in exams come up about stem cells, it usually talks about a range of ethical arguments. Some of these are that the cloning of human embryos is very contentious. Right? You need to be able to use your ideas about science to say clearly what the issues are. So, for example, human embryos will be destroyed during the cloning process. You may want to summarise different views that people have. So, for instance, some people may view it as murder. There are a range of other arguments to support it. So, if you're talking about treating people with neuron diseases or other diseases with the nerves, these could be treated using stem cells. The final area in this section is how genes are inherited. Genetic diagrams are used to show how the possible outcomes of a particular cross of a dominant allele is carried out. A dominant allele is shown by a capital letter and a recessive allele is shown by a lowercase letter. Cystic fibrosis is an inherited disorder caused by recessive alleles. The genetic diagrams that we're going to look at momentarily show the possible outcomes when both parents are heterozygous for the faulty allele. Heterozygous means that the parents have one dominant allele, so one gene that does not carry the disorder, and one recessive allele, so one gene that does carry the disorder. The genetic diagrams show the possible outcomes in both cases. So the top diagram shows if both mother and father contain, so the mother contains two dominant alleles and the father contains one recessive allele. So in this case, all four of the possible outcomes all have a dominant allele, so none of them will be able to develop cystic fibrosis. Underneath is where the mother and the father are both heterozygous, having one dominant allele and one recessive allele. In this case, 1 out of 4, or there is a 3-to-1 ratio, or there is a 25% chance that one of their offspring may develop cystic fibrosis. Thanks for watching. Remember, start revising earlier. Quality revision is better than quantity revision. Keep asking yourselves questions. Follow the link below for a summary sheet that covers many of the things that we've talked about in this video. Good luck with your additional biology exam.